So it's now time to move on and to introduce our second speaker, who is Dr. Yitan Zhu. Is a senior computational scientist in the Data Science and Learning Division of the Argonne National Laboratory and a senior scientist at the University of Chicago Consortium for Advanced Science and Engineering. His research interests include machine learning, deep learning, cancer genomics, drug discovery, and medical informatics. Before joining the Argonne National Laboratory, Dr. Zhu worked in the pharmaceutical industry and hospital organisation. For the past 15 years, Dr. Zhu has been working on developing machine learning and statistical methods to integrate and analyse biomedical data for disease research and therapeutics development. He obtained his PhD from the Department of Electrical and Computer Engineering at Virginia Tech, and he also holds a master's degree in patent recognition and intelligence systems. So thanks very much, Dr. Zhu. If you can get your slides up, and we look forward to hearing from you.
Okay, thanks for the introduction. Good day to the audience. I'm going to share my slides. I think you can see my slides now.
That's all good.
Okay, great. So this talk is about the computational prediction of anti-cancer drug response in preclinical drug screening experiments using machine learning and deep learning approaches. I am from the Argonne National Laboratory. Argonne is a research facility of the US Department of Energy. You might be a little surprised to know that Department of Energy lab is working on cancer research. So here is some background information. In 2016, the US Department of Energy and the US National Cancer Institute engaged in a strategic interagency collaboration to simultaneously accelerate advances in precision oncology and scientific computing. The Department of Energy wants to use its computational facility and analytical expertise to help drive cancer advances. The National Cancer Institute wants to use its cancer research resources and topics as a use case to help drive computing advances. The DOE and NCI collaboration is part of the Cancer Moonshot Initiative. Our Argonne National Laboratory participated in the collaborative project since the beginning. We worked with the NCI on computational drug response prediction. The collaborative research project is divided into two phases. The first phase is called Pilot 1 because we had 3 pilot projects at that time, each focusing on a different aspect of cancer research. The Pilot 1 project focused on developing new novel drug response prediction models. The second phase of the project, which we call it Improve, which is our current project. The focus of the Improve project is to develop a benchmark, comprehensive framework to evaluate and compare existing drug response prediction models. To support the collaborative research, we also built 2 infrastructure projects, Candle and MODAC. Candle is a distributed deep learning environment that we built to support large-scale runs of cancer computational models on clusters and supercomputers. MODAC is an online platform for storing and sharing data and models developed in the collaborative research projects. Our general research goal for this project is to develop machine learning models to predict anti-cancer drug response to optimize preclinical screening of drugs. The cancer models that we usually use in the preclinical drug screening experiments are immortalized cell lines, patient-derived xenografts, PDXs, and patient-derived organoids, PDOs. We are using the molecular data and the compound screening data of cell lines, PDXs, and PDOs, and also compound information to build machine learning models to predict drug response of drug screening experiments. Then based on the prediction result, we can select the experiments that are most likely to give a responsive result for future experiments. In this way, we can help to improve the cost efficiency of drug screening studies. We take the drug response prediction problem as a function. The input parameters of this function is tumor representation, drug representation, and meta information if available. This can be patient treatment history or other information. Then the output of this function is drug response. For the tumor representation, we can choose various types of data to represent tumor, such as the gene expression, mutation, protein expression, DNA methylation, or even pathology images. For the drug representations, we can also use different types of data. For example, the numerical descriptors, binary fingerprints, or the graphical representation or smile screens. If the drug response prediction task is dose dependent, we can also input doses as the input parameters. For the output of this function, the drug response, it can be measured by various different efficacy metrics, such as the half maximum inhibitory concentration, IC50, and the area under the dose response curve, AUC. In the past few years, we made quite a few technical achievements, which are pioneering works in this field. We developed a deep learning model to predict the response of drug combinations. This model achieved a coefficient determination R² equal to 0.944 in 5-fold cross-validation for the dose-dependent drug response prediction. So the picture here shows the architecture of this deep learning model. It uses multi-omics data of cell lines and drug descriptors to make prediction. Each type of input data is encoded by its corresponding neural network layers. Then the embeddings from the multiple data types are concatenated and then forwarded to fully collected layers with the residual collection to make prediction. We also conducted a transfer learning study for building response prediction models across drugs. In this study, we demonstrated that ensemble transfer learning can leverage the prediction patterns learned on large drug screen datasets to improve the prediction performance on relatively small drug screening study. We also conducted the first large-scale learning curve analysis for drug response prediction. We usually call drug response prediction as DRP for short. In this particular study, we showed that with deep learning approaches can outperform traditional machine learning methods for drug response prediction when the training data size is larger than 40,000. We also conducted a comprehensive investigation on model generalizability between different drug screen datasets. In this study, we discovered that the differences in viability assays can limit model generalizability between datasets. For example, if you have two datasets which are generated using the same viability assay, a model that is trained on the one of the dataset is more generalizable to the other dataset than the case that if the two datasets are generated using different viability assay technologies. In this study, we also discovered that drug diversity is more crucial than tumor diversity in the training dataset for raising the model generalizability across datasets. For this talk, I will talk about 3 of our research projects. The first one that I will talk about is the image generator for tabular data, IGTD. It is an algorithm that can convert gene expressions and drug descriptors of table format into image representations. Then we can use convolutional neural network models to model drug response based on the image representations. In the second project, we use data augmentation and multimodal learning techniques to predict drug response in patient-derived xenografts, PDX, using gene expressions and pathology images. The last project I will talk about is our current project, Improve. The goal of the Improve project is to develop a community-based automated framework to perform large-scale evaluations and comparisons of DRP models. Our purpose is to fill the vacancy in the field that there is currently no benchmark or consensus on evaluating DRP models. So the first section is about the IGTD algorithm. Many drug response prediction models are built based on cancer omics data and drug feature data. By default, these two types of data are in table format. Convolutional models have been recently very successfully used in many applications, where important information about data is embedded in the order of features, such as speech and imaging analysis. Because the convolution kernel and convolution layers can well capture the spatial relationship of features in the data to facilitate the prediction. On the contrary, tabular data usually do not assume a spatial relationship between features, and thus are not ideal for modeling using CNNs. To apply CNNs for drug response modeling, we develop image-generated tabular IGTD algorithm to convert the gene expression profiles of cancer cell lines and the molecular descriptors of drugs into their respective image representations, one image for each cell line or drug. The algorithm first assigns features to pixel positions so that similar features are close to each other in the image. Then, as illustrated by the picture here, for each column in the tabular data, which is a sample, the algorithm will generate a heat map to represent the sample. The intensity of a pixel in the image shows the value of the feature, of the corresponding feature in the sample. The code of IGTD algorithm is freely available on GitHub. You can use it for your own analysis task. Not necessary drug response prediction because IGTD is a general algorithm that can be applied on any tabular data. I will briefly introduce the IGTD algorithm. The first step of the algorithm standardizes features and calculates pairwise distances between features. The example here includes 2,500 features. We rank the feature distances and give high rank values to large distances. Then we use the rank values to generate a distance rank matrix between the features as shown in figure A here. So this matrix is a 2,500 by 2,500 matrix. The black elements in the matrix indicate high rank values, large distances. Because we have 2,500 features, naturally, we want to convert them into a 50 by 50 image. The second step of the algorithm calculates pairwise distances between pixels based on their grid locations in a 50 by 50 image. We also rank the pixel distances and give high rank values to large distances. Similarly, we can generate a pixel distance rank matrix as shown in figure B here. It is also a 2,500 by 2,500 symmetric matrix, but it shows some interesting visual pattern because the pixels are concatenated row by row from the image to form this matrix. You can see the top right and the bottom left corners are darker because there are distances between pixels from the top rows and the bottom rows in the image. So the distances are large and the rank values are large. The region around the diagonal is brighter because there are distances between pixels from adjacent rows in the image. So the distances are smaller and the rank values are smaller. And the whole matrix shows a mosaic pattern. The small tiles in this matrix correspond to the pairwise combinations of rows from the image. I copied the feature distance rank matrix and the pixel distance rank matrix from the previous slide to this slide. They are figure A and figure B here. Then to assign features to pixel positions in the image, we can simply assign the eyes feature in the feature distance rank matrix on the left to the eyes pixel position in the pixel distance rank matrix on the right. Then to assign similar features to adjacent pixels in image, we can modify the order of the features in the feature distance rank matrix so that it becomes similar to the pixel distance rank matrix. To measure the difference between the two matrices, we define an error term to measure the difference. It is basically a summation of all element-wise differences in the two matrices. The IGTD algorithm searches for an optimized assignment of features to pixel positions by iteratively swapping the positions of two features in the feature distance rank matrix to minimize this error. Figure C here shows the feature distance rank matrix after optimization. You can see it shares the visual patterns of the pixel distance rank matrix. In figure C, you see the top right and the bottom left corners are darker. The diagonal region is brighter, and also the matrix shows a mosaic pattern. Figure D here shows the reduction of error over time. After about 5,000 iterations, the error starts to converge. A noteworthy thing here is when we switch the positions of two features in the feature distance rank matrix, we need to switch the corresponding rows and columns of the two features simultaneously in the feature distance rank matrix. Then we can use the IGTD algorithm to convert the gene expression profiles of cell lines and the molecular descriptors of drugs into their respective image representations. Figure B here is an example of the cell line images. In this heat map, the intensity of a pixel indicates the expression level of the corresponding gene. In figure C, you see a heat map of a drug. The intensity of a pixel indicates the value of the corresponding drug descriptor in this drug. After converting the cell line expression profiles into images and the drug molecular descriptors into images, we can use the convolutional neural network to model drug response based on those image representations. The flowchart here shows the architecture of this convolutional neural network model. It has two sub-networks for the input of drug images and cell line images separately. Each sub-network has 3 convolution layers with batch normalization, radio activation, and max pooling. The embeddings of these 2 sub-networks are concatenated and forwarded into dense layers with dropout mechanism to make prediction of drug response. We test the CNN modeling with IGTD images on 2 benchmark models, benchmark datasets of drug screening. These are CTRP and GDSC. We measure the prediction performance using R-squared, the coefficient of determination, and we compare the prediction performance to those of 4 baseline prediction models that are trained on the original tabular data. These include 90 GBM, a gradient boosting machine, random forest, and 2 deep neural network models. The result here indicates that our CNN modeling with IGTD images statistically significantly outperform the other 4 prediction models that are trained on the original tabular data. So we also think about why CNN modeling with IGTD images can outperform. We think our datasets where the physical relationship between features can be characterized by feature similarity, the IGTD images can better represent the feature relationship, which can be captured by the single model to make prediction. For example, in the drug response prediction problem, the co-expressed genes may work together in one functional gene module that is involved in cancer mechanism and treatment mechanism. This can be captured by the single model to facilitate drug response prediction. In this section, I will talk about the construction of DRP models based on PDX data. So far, almost all existing drug response models are constructed based on cell line drug screen datasets. The reason is simple, because cell line drug screen data are relatively abundant. Some very large dataset can include more than 300,000 of experiments. On the contrary, drug screen data of patient-derived xenografts, PDX, are usually very small. A few hundred can already be a very good data size. But we all know that the PDX are much better representations of patient tumors than immortalized cell line, because they provide in vivo environments for the tumors to grow. We conducted a feasibility study to investigate building drug response prediction models based on PDX gene expressions and pathology images. The picture on the left shows the process of developing PDX models and using them in drug screening experiments. The primary tumor specimen is first extracted from patient. Then the tumor cells are implanted into immunodeficient mice. The tumors will grow, and after some time, the tumor tissues will be collected and implanted into more immunodeficient mice. Then we will have a family of PDX passage after passage. All of the PDX models in this family is derived from the same original patient tumor. And in the drug screening experiments, some mice of this family will be treated by vehicle and used as control. Some mice will be treated by a single drug treatment, and some mice will be treated by drug pair. For this particular study, we use an unpublished PDX drug response data from the National Cancer Institute Patient-Derived Models Repository, PDMR. So the PDMR PDX experiments are well designed. They include usually 10 mice in each of the control and the treatment groups, so we can get a very reliable response measurement. The picture on the right here shows the median tumor volume growth curves of one treatment group, of one control group, and 3 treatment groups. The green curve, drug B, indicates an effective treatment because it can reduce the tumor volume. And the red curve, for example, the drug C, is a treatment that is not very effective. Based on the tumor volume growth curves of control and treatment groups, we have an expert from PDMR to make a call on response versus non-response. In this data set, the tumor data include gene expressions and pathology images of multiple mice in each PDX family. By PDX family, we refer to the multiple mice that are derived, multiple PDX models that are derived from the same original patient tumor. We have gene expression profiles and the whole slide images of 487 PDX models belonging to 96 different PDX families. For the gene expression data, we don't use the full transcriptome data for the analysis. We use the expression values of less than 1,000 landmark genes, which are selected by the LINX project. The links genes have been demonstrated to well infer the expression variations of more than 80% of all transcripts. For the whole slide images, we had a pathologist to annotate the tumor region of interest on the slide. Then the tumor region is tessellated into tiles and the background tiles are removed. To represent drug, we use the Dragon version 7 software package to calculate the molecular descriptors. Totally, we use close to 2,000 descriptors. They indicate various types of chemical properties, for example, atom types, estimations of molecular properties, topological and geometric descriptors, and many other kinds of properties. The response data are generated by 12 single drug treatments and 36 drug pair treatments. We have close to 1,000 experiments. Those are unique combinations of PDX families and treatments. About 5% of the experiments are positive, responsive. The others are non-responsive. To form the data used in this analysis, we first match the response value in each experiment with the multiple pairs of gene expression profiles and pathology images of the PDX family used in this experiment. Then we performed 2 data augmentation steps. In the first step, samples of single drug treatment has their drug features duplicated to become a pseudo drug pair treatment. The purpose of doing this is to combine single drug treatment samples with the drug pair treatment samples for analysis. In the second data augmentation step, the samples with drug pair treatment have the positions of the 2 drugs switched to generate an additional pseudo samples. Then all the samples from the same experiment are taken as one treatment group. So the picture on the left illustrates the data augmentation process. For the first experiment, which is group 1, we have K pairs of gene expression profiles and pathology images. So we have K samples here. And because this is a single drug treatment sample, we duplicate the features so that it becomes pseudo drug pair treatment samples. For experiment M, we have N pairs of gene expression profiles and pathology images of the PDX family used in this experiment. And because it is a drug pair treatment experiment, we switch the positions of the 2 drugs and to generate additional pseudo samples. So the sample size is doubled. You see 2 N samples here. Then we use a multimodal neural network model, MMNet, to model the drug response. It has 4 components. For the inputs of features of 2 drugs, 1 gene expression profile, and 1 histology image tile. We use the convolution layers from the exception neural network model to encode the histology image tile. The exception model is pre-trained on ImageNet data. The embeddings from the 4 data inputs are concatenated and forwarded to dense layers to make prediction. We compare the drug response prediction performance of MMNet to those of 3 baseline model architectures. The first baseline model is UME-NET. It is a neural network model, uses gene expressions and drug descriptors for making drug response prediction. It takes the same model architecture as MMNet, but has the 4th component corresponding to histology image tile removed because it doesn't use the image for prediction. The second baseline model is UMH-NET. It is a neural network model using histology images and drug descriptors to make prediction. It takes the same architecture as MMNet, but has the 3rd component removed, which is corresponding to the gene expression profile because it does not use gene expression. The third baseline model is LightGBM, a gradient boosting machine model that uses gene expression and drug descriptors to make prediction. We evaluated the performance of these models through cross-validation and use 3 different performance metrics to measure the prediction performance. This includes the measured correlation coefficient MCC, area under the receiver operating characteristic curve AUROC, and area under the precision recall curve AUPRC. The table here shows the prediction performance of all the prediction models. A character V in the table means a certain type of feature or samples are used to build the prediction model. The last 3 columns in the table shows the prediction performance of all the models. We include 2 more models here, which are UME-led original and UME-led pairs. These models take the same architecture of UME-led, but they are trained on not fully augmented data. The purpose of including them here is to compare with the UME-led model, which is trained on the fully augmented data. We want to see whether data augmentation techniques make any difference in drug response prediction performance. The result shows that our data augmentation techniques indeed statistically significantly improved the drug response prediction performance. Then we further compare the prediction performance of MME-led with UMH-led and UME-led. The purpose is to see whether the combination of pathology images and gene expressions can outperform using gene expressions or pathology images individually. Our result indicated that the combination of the pathology images and gene expressions can statistically significantly outperform using pathology images only, but not reliably outperform using gene expression only. The next project is our IMPROVE project. It is our current research project. Every year, we see more and more DRP models developed and published. The bar plot on the left shows the numbers of deep learning DRP models published in the recent years. They use different software frameworks, among which TensorFlow and PyTorch are the most popular. The bar plot on the right shows the number of deep learning models belonging to different categories. You can see they use different drug representations, different cancer representations. The models use various deep learning architectures and schemes. When evaluating the models, the model developers use different evaluation methods and compare their models to different baseline models. So although there are so many DRP models developed, you can see there is no consensus about many things in this field. For example, we don't know which drug representation or cancer representations are more predictive. There's no consensus on what deep learning approaches produce more generalizable models. And also there's no consensus on how to evaluate models and what data should be used for evaluating models. All these observations motivated our IMPROVE project. The full name of the IMPROVE project is Innovative Methodologies and New Data for Predictive Oncology Model Evaluation. Our purpose is to fulfill the need that whenever a new DRP model is developed, it needs to be evaluated and compared with all previous models. So we want to know which models are performing better and what are each model's relative strengths and weaknesses. This includes the following. For example, we want to determine what aspects of the model formulation, structure, training protocol can make a difference in performance. We want to understand training data impact on performance. We want to determine the types of errors a model is making. Also, we want to evaluate model generalization in tumor space. This is a typical precision oncology application case in which we want to know how well the model can make prediction for a new cancer case that has never been seen before. Then we also want to evaluate the model generalization in drug space. This is a drug development application case in which we want to evaluate how well a model can make predictions for a new drug that is not included in the training set. Also, many models use prior biological knowledge in their modeling process. We want to know whether those models can effectively use those prior knowledge in their modeling process. So our general research goal is to build a community-based automated framework to make massive evaluations and comparisons of DRP models feasible. The improved framework will include a good number of public DRP models for evaluation and comparison. The table here shows the DRP models that we are currently working on. For the current phase of the project, we set up some model selection criteria to include models into our framework. We are working on deep learning models. We focus on general models that can make prediction across drugs. This means the models that can make prediction for drugs that has not been seen by the model before. For the current phase, we focus on only drug models that make prediction for single drug treatment. Apparently, the code of the model needs to be available, and also we require sufficient technical documentation to utilize the model. Currently, we are curating more than 20 DRP models. The model curation step includes many things. The first step is to retrieve the model code, debug it, and test the training and the inference of the model. The second step is to reproduce results in the publication if the data used in the paper are available. Then we need to standardize the model code for large-scale runs. We need to restructure code into 3 modules. Model training for building the prediction model. Model inference for using the model to make prediction. And the data preprocessing to prepare input data of the model. We need to standardize the input-output interfaces of each module across models. So all the models need to accept the same data format. This includes not only the input data of the model, but also the output data from the model, such as the prediction outcome. We also want to make the models compliant with our Candle platform by adopting Candle parameters and functions. Candle is a distributed deep learning environment that we developed for supporting large-scale runs of models on supercomputers and clusters. The last step of the model curation is to generate a singularity container of the model to further improve the compatibility and the portability of the code. Large-scale runs are necessary for using the improved framework to evaluate a DRP model. The reason is threefold. First, our improved framework will include multiple evaluation workflows. Each of the evaluation workflows will assess one aspect of a DRP model. Then each evaluation workflow requires many analysis runs because, for example, for the cross-validation, we need to conduct many times for model training and model testing. Then in each analysis run, we need to do hyperparameter optimization, which still requires many runs for training a prediction model and testing a prediction model. So the total number of times that we need to train a prediction model can easily reach the scale of hundreds of thousands. I include here a screenshot of our improved project on GitHub. It has more than 40 code repositories. Many of these code repositories are the curated DRP models. We forked their code from their original sources and modified their code here. We will implement multiple model evaluation workflows in the improved framework. The first model evaluation workflow we want to implement includes multiple cross-validation schemes within a dataset. The figure on the left here illustrates the multiple cross-validation schemes. In each table, the columns are cancer cases. The rows are drugs. The yellow circles are testing samples, and the blue circles are training samples. In the first cross-validation scheme, which is a mixed set, the training set and the testing set can share both cancer cases and drugs. In the second cross-validation scheme, which is cancer blind, the training set and testing set cannot share cancer cases, but can share drugs. In the third scheme, they can share the cancer cases, but not drugs. In the last scheme, which is called disjoint set, both cancer cases and the drugs cannot be shared between the training set and testing set. The table on the right here shows an example result of another cross-validation, another model evaluation scheme, which we call cross-study generalization analysis, CSG. The CSG analysis is to assess the generalization of model across different drug screening datasets. So in this table, we have 5 cell line drug screening datasets. The workflow will train a prediction model on one dataset and test its prediction performance on this dataset and all other 4 datasets. Then the prediction performance will be filled into this table. After the whole table is completed, we can clearly see the generalizability of the models between the datasets. We will also implement a learning curve analysis workflow to assess how the model performance changes with more training data. In this workflow, a model will be trained with multiple training data sets with different set sizes. Then, as illustrated by the picture here, we can draw a curve of the prediction error in terms of training set size. So the analysis will study the data scaling property of models. We may discover that some models can improve their prediction performance more quickly than other models when the training set size increases. Another model evaluation workflow we want to implement is the model robustness analysis. The purpose is to investigate model performance in variations of inputs and evaluate its sensitivity to changes in data and the model training scheme. For example, we can inject noise into the drug response data and then observe how the prediction performance drops when more noise is injected. We also want to implement a post-prediction error analysis module in the improved framework. The purpose is to summarize prediction error uncertainty specific to cancer cases, drugs, cancer types, and drug categories. Besides the curated models and the model evaluation workflows, we also need to have benchmark data to support the model evaluation and comparison. We designed a unified data schema to organize data from different sources. For example, a cell line or a drug is always represented by a unique name across the datasets. The flowchart here shows the unified data schema and how the various data are organized and processed. Each block here indicates one type of data. The data type name is on the top with the data fields in the data table shown following the name. The meta information of cell lines is retrieved from Celosaurus and CCLE. CCLE stands for the Cancer Cell Line Encyclopedia. Then the multi-omics data of the cell lines is retrieved from DepMap data portal of CCLE. This includes the gene expression, DNA methylation, copy number mutation, protein expression, and microRNA expression. We retrieve the meta-information of drugs from PubChem, and based on the SMILE streams, we can calculate the numerical descriptors and fingerprints of every drug. We obtain the response data of 5 cell line drug screen datasets, including CCLE, CTRP, version 2, GDSC, version 1 and version 2, and GCSI. Then based on the viability readouts of multiple doses in an experiment, we can calculate multiple dose-independent efficacy metrics, such as the AUC and IC50. The last piece of data is generated by the improved pipeline. These are the data partition files. They specify the samples that should be included in the training set, validation set, and testing set in each analysis run. Then all the data pieces are integrated and matched together to form the benchmark data for model evaluation. So everything in our improved project is open and online. We have a GitHub project to host all the code, including the repositories of curated DRP models, the model evaluation workflows, and the APIs, and also relevant resources, for example, the scripts for data curation and processing. We also have a website to introduce the improved project and the curated models. On this website, we provide introduction and guidelines of curated models, and also tutorial for making models compliant with Candle and generating synchronistic container. We have an FTP site to host data used by the improved framework and the curated models. To summarize this talk, we first give an overview of DOE NCI collaborative research on anti-cancer drug response prediction. Then we talked about our image generator for tabular data algorithm, IGTD algorithm, for converting data into image representations for modeling using convolutional neural networks. We talked about the construction of DRP models based on patient-derived xenograft, PDX, gene expression, and pathology image data. Lastly, we talked about our currently improved project to build a benchmark framework for automated and comprehensive evaluations and comparison of DRP models. These are the references used in the talk. I want to thank my colleagues at the Argonne National Laboratories and our collaborators from the Frederick National Laboratory, Mayo Clinic, National Cancer Institute, University of Nebraska, Texas Tech University, University of Chicago, Los Alamos National Laboratory, and Pacific Northwest National Laboratory. This is the end of my talk. Thanks for your attention. I'm happy to take any question.
Thanks very much, Dr. Zhu, for that fantastic talk. I must say it's really impressive to see all the work that you're doing, bringing together existing models and rationalizing them and making it possible to compare them. That sort of work is really useful to the community, and I think probably often quite difficult. Is that correct?
Thank you. Yeah, yes, it's especially for the improved project. It's a very large project. We need to curate a lot of models from the public domain and standardize them and run big pipelines on supercomputers. So we have a big team working on that, probably more than 20, close to 30 people.
Yeah, it looks like a lot of work.
Yeah.
Yeah. So looking in the Q&A here, we have a question from Fred Fung. He said, can he ask questions regarding the Candle ECP project, which I came across a while ago? I recall back then I read somewhere which mentioned there were challenges of model parallelism in the Candle project. I wonder how it goes so far. It says, I also remember Candle was shipped as a native build on the Supercomputer Summit instead of inside a container. My second question was why to use that approach. So I guess the first question is, what were the channels of model parallelism in the Candle project? Challenges of model parallelism in the Candle project?
For the Candle project, the Candle project actually has two components. The first component is the Candle library. The second component is the supervisor workflow. The Candle library defines a lot of input and output parameters and functions that can be used to support running a prediction model. So the Candle library basically standardizes a model. And then after the model is standardized, the Candle supervisor part can automatically submit the models and including their input parameters to run large-scale analysis on supercomputers. And it can also actually collect the information output from the models to automatically organize the future batches of the analysis. For example, for the hyperparameter optimization, usually you can launch one batch of analysis, then you can get the validation loss of the model training to select the next set of parameters to be searched in the optimization process. Hope this answers the question. And what is the second question?
Yeah, thanks very much. Yeah, the second question was regarding, sorry, Fred Fung's from the NCI Australia. Sorry, I've got the wrong one. They said the Candle was shipped as a native build on the Supercomputer Summit instead of inside a container. Why use this approach?
Oh, yeah, so the Candle workflow does not require a container, definitely not. You can just build your Python scripts, and the Candle supervisor, the workflow, can take the scripts and automatically to add the input parameters and point it to the data files, and it can submit a large batch of jobs to supercomputers. The container thing is a one step of model curation in the Improve project, because we want to further standardize everything. And because those models are from the public domain, building a container helps us to standardize things, and we don't want to change anything in the large scale runs once we have the code ready. That's the purpose of using container here.
Thanks very much for that answer. Can I just ask myself, with your model that uses the method to generate the images, and you incorporate information from the tumor and from the drug itself as in an image format, can you work backwards and use the information in the images to provide information about what drugs are effective? Can you back information about the drugs themselves back out of those models?
Do you mean once we use the model to make prediction based on the image representation of cell lines and drugs, can we infer back which feature of drug is important?
That's correct, yeah.
Okay, so that's usually a quite a challenging problem for deep learning model, because the deep learning model is very complex, not like the conventional machine learning methods. You can, for some conventional machine learning methods like random forest and 90 GBM gradient boosting machine, you can easily trace back which feature is more important. But recently, there are also some methods developed that you can examine which feature in the deep learning model can give a higher weight than other features for your prediction. Like if I remember the name correctly, it's lift over or something. And also there are some packaging that can help to calculate Shapley values of those features, indicating the importance of the features in deep neural network model. There are some papers on that.
But in general, it's not easy.
Yeah, for deep learning model, usually it's not that easy because the model is too complex.
Yep. Okay, thanks very much for your presentation today. We look forward to seeing you again in the Q&A session at the end of all the talks. So
thanks for having me here. It's my pleasure to give this talk.
Thank you.